waiting for the few more people to come from the open mic sessions, but before that, we are getting ready for a fireside chat. Before that, just to get some things moving, we also have a small activity right now. So, I think we spoke about this at the start of the day, but I want to bring it back again, that one of the things that I do is I do quizzes as part of my science communication. And some of the things that I do is ask questions which relate to a certain theme, or so that students and even young adults can figure out a story, specifically in history of science, behind very interesting things. Now today, at random times in the day, I'll be asking certain questions and you will get a chance to win a prize. So here's how it's going to go. All my questions are about women who have changed the world in science. But I'm going to be asking questions in a very easy, informal way. I'm going to mention something, probably a name or an object or an entity, and you have to put up your hand and tell me who is the lady behind it and in what way she changed history or she changed science, right? So it's very simple. I'm going to say something out and you have to tell me the name of a woman who's changed history or science through the work that she's done. Now, can you have the lights down, please? Because I need to see who's putting up their hand. So if you think you know the answer, put up your hand. Oh, you are, wow. I haven't asked a question yet. I like your confidence. So I'm going to say a word and you got to answer it. If you get it right, we will give you a prize. And for your confidence, I'm going to look at you. And so I'm just going to say pitch blend. Pitchblende. That's the word. So who is the lady who changed science and the history of science thanks to her work with pitch blend, Pitchblende. We have a gentleman up there who's put up his hand. Hi, if you could just stand up, wait for a second till the mics turn green, introduce yourself and then tell me the answer. Yes. Harsh, can we go? Audience mic, please. Yeah, we're waiting for it to turn.
Yes, Marie Curie, because she worked with pitch blend and she isolated uranium from that.
Okay. And in what way did she change history or change science? You know,
basically, it was the first time found out radioactivity came out. So,
okay, now my first question to you was, please introduce yourself. So we do not know your name or where you're from.
Yeah, so I'm Ameya, I'm from Hyderabad.
Ladies and gentlemen, can we have a round of applause, Ameya? Well done. So, yes, the word I gave was pitch blend, and Marie Curie worked on pitch blend to give us two elements, radium and polonium, and then she started off a trend in a family which is has put Indian families to complete embarrassment by starting a family that won 5 Nobel Prizes. She and her husband won 2, then her daughter won another, then her son-in-law won another, and then they had another one in the family just for fun. So yeah, my mom and dad are not going to be happy with any prize that I win after that. But yes, that is the Marie Curie family, the Curie family and the 5 Nobel Prizes. Well done. Okay, so second term, here is your entity. It's called Photograph 51. Oh, right in the back, there, full speed. Excellent, yes. Can you introduce yourself and tell us what the answer is?
My name is Mrinal. I am from NCBS Bangalore, and photograph 51 was taken by Rosalind Franklin, and it's an X-ray crystallographic picture of the structure of the DNA, and that was a very important picture that helped Watson and Crick reduce the structure of DNA. And they kind of say there was hard.
Yeah.
I mean, they say it was stolen from her and she didn't know that they took her.
No, they didn't say and all. That is absolutely true. Nothing of that. Those who were drinking in Cambridge in a pub called the Eagle Pub. If you go to the Eagle Pub, which is in Cambridge, you walk there, they have a plaque which says, this is the place where Watson Crick was sitting when they had, no, they were drinking. They brought, and they took the photograph from Rosalind Franklin's table, showed it to them, and that's how they got it. Yes, well done, Mrinal, you also get a prize. That is obviously Rosalind Franklin, without whom I wouldn't know who I am, because DNA. Yes, double structure. So, excellent. So, let's get the Fireside chat up and running. And it's absolutely my pleasure to welcome on stage fireside chat about spillover stories, infectious diseases, and zoonoses. May I call on stage Sunit Singh and Joel Joseph.
All right. Welcome everyone once again to this fireside chat on spillover stories, infectious diseases and zoonoses. I am Joel P. Joseph. I am a freelance science writer / journalist and also a researcher based out of Bangalore. And with me is Sunit K. Singh. He is currently the director of Dr. B.R. Ambedkar Center for Biomedical Research at Delhi University, and also has additional charge as director of the Delhi School of Public Health. He is a very renowned molecular virologist, specializing in emerging infectious diseases, vector-borne viruses, and zoonoses. And so we're going to hear a lot about infectious diseases and zoonoses from him. I met him just this morning, which I think is a feature for a fireside chat and not a bug. And I'm going to offload a lot of questions on to the audience. And also, incidentally, just before walking up stage, I heard that today is World One Health Day. What a day to have a fireside chat such as this. All right, with that introduction, so I'll just brief you a little bit about the structure of how we are going to follow this. I know that this is a really vast topic, and you can go for days, and we have people who are able and experts in the field who can take this conversation for days, but we only have 40 minutes or below that now. So we're going to discuss a little bit about endemic viruses that have pandemic potential in India, a little bit about zoonotic spillover events, and then how we as journalists can communicate zoonotic events, outbreaks, and provide solutions journalism in that space. And we'll open the floor for questions. As we begin, Sunit, give us a 30-second or a 60-second primer on what infectious diseases are, what we mean by vector-borne viruses and zoonoses, and what piqued your interest in this area and how you got to where you are today.
Thank you, Joel, and thank you to Shubhra and SJAI team for inviting me over here. So first I will answer the last thing which you asked me. How did I end up in working in infectious diseases? So I still remember it is taking me few years back when I joined CCMB Hyderabad as a faculty. And then director of that institute said, what's your postdoctoral training? I told it's Borrelia burgdorferi, another zoonotic disease that is reported that causes Lyme disease and is well known in Europe and USA. He said, come on, Sunit, you need to stop all this and you have to do whatever is the relevant for India point of view. So this is the beginning which, put me in the infectious diseases, and I started my career from HIV. In a way, yes, you can say it's also a kind of, comes under that category, zoonotic. Since then, I am working on various vector-borne diseases. So vector-borne diseases are those diseases which require a kind of either a organism to transfer from one place to another. Take example, Japanese encephalitis, dengue, chikungunya. All these require mosquitoes, and there are different species of the mosquito. But when we go towards Crimean Congo fever, even Kyasanur forest disease, these all are transmitted by ticks. So these are arthropods. So we give another terminology to them. We say them arboviral diseases. So these are arboviral diseases which are known in this country, and some of them even increasing its geographical area from one place to another place based on various changes.
Thank you very much. So, just following up on that question that you first faced when you came to India. So, what are some of those viruses with pandemic potential in India, and some of those that you see zoonotic outbreaks of frequently from your experience?
Very good question. So, as you said, after my joining, it was Japanese encephalitis. So there is a region in eastern Uttar Pradesh that is known as Gorakhpur. And our chief minister of UP also comes from that place only. So one day I saw a heading in Indian Express, and that was this: a season of death. So this season of death was something like this. A small story is behind that. There was a family who had 3 sons, and one of them got, fever, high fever, headache, and lot of pain in the body. And the father took him to the nearest medical college, that is the BRD Medical College. And then what happened? He got admitted over there. And as soon as he reached to the hospital, he got another phone call from family. Second one is also facing the same kind of symptom. As soon as he rushed to pick up second one and came, first one was died. And same happened with the 3 kids. So there was a issue whether it is viral disease, whether it is non-viral disease, or there is a phenomena also reported in that area that during lychee season it happens. You would have read these stories. It might be due to lychee. Then later on it was found most of them is related with the Japanese encephalitis virus infection, and which is transmitted by Culex mosquito. And yes, few cases were there. That's why it was clubbed together under acute encephalitis syndrome, AES. Few of cases were there where enterovirus was reported. That is not a kind of vector one. So it was found during that area, sanitation practices were not very good. So what used to happen, people used to go to open areas for defecation. And water level of that area is quite high. That is, in other words, we say Tarai area where water level is very high, very famous for paddy farming. So what used to happen, that fecal matter used to get mixed with the water which people used to for drinking purpose, and then they used to get enteroviral infection. But yes, most of them was Japanese encephalitis virus, which is transmitted by Culex mosquito. Yes.
So, are there other viruses that also cause encephalitis? And if you could explain a little bit more on that.
So, when we say encephalitis, yes, so most of us will not be knowing probably even dengue virus causes encephalitis, even chikungunya virus causes encephalitis, and we have reported in our laboratory study how dengue enters from peripheral systemic infection to the brain. So that uses a protein, we reported already published, which leads to results to the dysregulation of tight junctions protein of the blood-brain barrier. Blood-brain barrier is a barrier which does not allow to move everything freely from peripheral circulation to the brain, because that might be dangerous for the brain. So this is so dengue is one of them. And normally, when we talk about dengue, this is a very special virus, because our immunology concept says once you will be bitten by a mosquito, suppose you got dengue, and then when next time you will be bitten by another mosquito having dengue, you should have a protection because you would develop antibodies. But dengue is having 4 serotypes: serotype 1, serotype 2, serotype 3, and serotype 4. The problem is here, once you got infected by one serotype and again you got infected by the same serotype, you will be protected. But once you got the infection by serotype 1, there will be antibody development against serotype 1, but soon after, if you got infected by serotype 2, those antibodies will not be protective, will not be neutralizing antibody against dengue serotype 2. So what will happen, that will bind with the, binding will be there, but that binding will be a non-neutralizing kind of reaction. So that would be taken up by macrophages. So rather than protecting from the dengue infection, the person would have severe pathologies and even infection will increase, and that terminology in biomedical science is known as antibody dependent enhancement, which should not be there. Then chikungunya also crosses the blood-brain barrier. Chandipura is very much reported near, Maharashtra. There is a place, Chandipur, and it was first reported there. And the mortality is close 60 to 70% if you got infected. It's endemic in that area. But now geographical area is increasing in a way. It is moving all the way from Maharashtra to Andhra and other places. Take example here in Karnataka, there is a Casanur forest disease. And Casanur forest disease is caused by Casanur forest disease virus. And this virus also leads to the encephalitis. So all of them have a kind of different mechanism of transfer, but yes, they end up to the same place, and that's the brain. Thank you.
Thank you for that very good explainer on how it works inside the body, how the disease develops in terms of the mechanisms that's involved. So when we talk about infectious diseases and zoonosis, we get a lot of stories on once it has been transmitted into humans. So there's multiple layers that we sometimes tend to miss, work that happens in the lab in understanding what kind of mechanisms are sort of driving this disease in humans as well as in animals, and then also what is that chain of events that lead to zoonosis, and also studying from the public health perspective of the community that is very closely linked to these some of these vectors that are present in the environment. So as director of institutions that work on all of these aspects separately, how do you basically bring all these silos together, and how can journalists see that story through that is taking place in different labs and in the community, and how they all play a role in coming together in communicating stories on infectious diseases and zoonosis?
So that's a very good question, quite long question, so I will go step by step. So as I was talking to all of you regarding the immunity, that's the way I define the integrity of ecological system is the first line of defense against zoonotic spillover. So normally when people talk about zoonotic infection, they are more concentrated towards biomedical aspect. But there is a big reason at the ecological interface. So what happens for any virus? Like take example, giving a very simple example. Suppose I came to Ahmedabad first time, I will take Joel's help where I need to go and which way I have to reach to airport or other places. He will tell. Means I need a source. Same way. These zoonotic viruses, it's very difficult for them to cross the species barrier. All the way from, take example, bats, just for example I am taking, bats to humans. It's not easy. It's not one-day job. Crossing species barrier have, as you said, different layers of complexity. First of all, there should be a contact in between the human population as well as these reservoirs. Once they come into contact, when we say spillover, it seems that spillover, they are always spilling over. It's not the case. What happens when we get kind of, take example, during a mother, when mother gets pregnant, there is a kind of immunosuppression. The same way, when you do deforestation, when you use the forest areas for urbanization, industrialization, for road construct, it's you who are invading into their territory. And once that land is invaded, the inhabitants of that land, who are these wild species, they undergo stress. As you have cortisol hormone, which is a very good predictor, okay, once it goes up, so yes, you are somewhere under stress. The same way it happens with the animals. Their cortisol also goes up. And what happens when if they are harboring any virus, and if those reservoirs go under stress, they also suffer from immune suppression, and that immune suppression gives chance to virus to multiply heavily. And once that multiplies heavily, what happens? Those animals start shedding off the viruses. And suppose you are entering in their territory, then you will be exposed to that. And suppose there are two kinds of inhabitants. Those who are inhabitants in that area, there are forest dwelling communities, they live. Slowly and slowly, they develop a kind of resistance. You might find antibodies if you do serology of those fellows. But if you are naive, your immune system has not seen that organism, you will have a very violent action from your immune system. And that action, it's not virus which will kill the persons. It's your hyperactivated immune response. What happened during SARS-CoV-2? There were different variants. When Delta came, very many of us, including me, was admitted into ICU because our immune system overreacted. So it's not Delta variant which killed many of the fellows around. It's only our hyperactivated immune response that led to endothelial cell permeability, hemorrhages, and that led to the, mass mortality. So we need to understand it here, that it's us who invade in their territory. And once we go close to them, boundaries are overlapping. You are prone to get exposed to those viruses. And once you get exposed, it's not always true that you will be infected. The reason for infection, that virus needs to enter into your cell, replicate there, and then your immune system will start, attacking, and then whatsoever happens. As I said, I will ask Joel, what is the way to airport? The same way, most of these zoonotic viruses, they try to find out a middle intermediate host close to the human population. So that will go all the way from, it might be or take example, influenza, that goes all the way from birds to swine. And that remains there for years and years. And swine is kind of domesticated by very many of us. And then what happens? Then there are chances of getting exposed. Now, your next part. What as a journalist one should do while reporting such kind of outbreaks? If you remember COVID outbreak, what happened? Most of them who had not even a single paper on virus, who never worked on virus, they were not knowing difference in between virus and bacteria, but overnight they became expert. For the sake of being kind of highlighted in TV or print whatsoever media. So what journalists should do, and here I think science journalists, I'm very happy here today, I said Sahana multiple times, because this was a deficiency in this country. We want to have science journalism. So a science journalist is able to recognize who is domain expert. Then domain expert, if you ask me, Sunit, why don't you speak on developmental biology? I want to speak. That's none of my domain. Being a biologist, I know what is developmental biology, but I do not want to become expert without having in-depth information. So it's very much required that we should approach right people at a right time to avoid misinformation and disinformation and help this society.
Very, very nice. With that note, I'll let the experts in the audience, who are the journalists, ask questions.
There is a relation between global warming and malaria disease. What's kind of relation and what do you think about this?
So when we talk about climate change and their effect on vector-borne diseases, so there are two schools of thought. One says, yes, there is a clear relation. Another says it's not validated, it's not proven till now. So what happens, I am just answering your question in terms of I have taken it general vector kind of. During higher temperature, it has been seen that it's very good for, so very nice question. So it's two aspects. One, as temperature increases, mosquitoes, replication of viruses and even mosquito thrive well. Their developmental stages go well. So I'm there, extrinsic incubation period. What is extrinsic incubation period? When a pathogen is within the vector, okay, that gets minimized. So that gets minimized, and even it has been reported biting rate increases. So there are more chances to get infected. But another question you will have, I am saying, yes, temperature goes up. Then I ask myself, how far? Suppose it got 40 degrees, then it would be good, because till 29 mosquitoes will work. That's why what is happening now, as our temperatures are going up, these vectors are moving towards higher altitudes, where there were no reports of such kind of infections, but now it's being reported there. So here it might reduce if it goes quite up, but those areas where native population exists and they are not exposed to such kind of vectors and infection, their situation might get worse. Thank you. Yes, please.
So you mentioned, the spillover when there is stress on the body.
we talk about mutation, so let me take a little bit back. So when we talk about viruses, there are two kinds of viruses. In general, one RNA and another DNA. RNA viruses are very prone to mutation. The reason, their photocopying machinery, I mean to say when we say replication, is error prone. Because the enzyme which makes their multiple copies is not having proofreading activity. So it is something more related with the kind of viruses. Take example influenza. It's quite, they have segmented genome. So what happens? There are various mechanisms. They can exchange their segments. Suppose a swine flu virus is there and a bird flu, and they are infecting in the same cell. Then as I said, they are having segments. They can exchange their segment, and that is known as a reassortment, a recombination event, and then a new virus. So what will happen? The antigenic epitope which our body was recognizing through our immune system against A and B virus will be ineffective here because a third type C virus generated by the method of recombination event. So it's a more in RNA virus.
We'll take one last question. We have only like 1 minute.
We are finding numbers of outbreak in different area in vector-borne diseases. Just like you explained, like Uttar Pradesh issue of that. So in most of the time there was a conflict, but we are not reporting, or you from government side there no proper reporting of that proper news. Where that news are not providing the general community. How to solve this issue?
So for this, yes, question is very nice, but for this there needs to be effective surveillance, okay? Normally what happens, we start surveillance program when outbreak is already there. There is no reason to do it, it's already there. Better to have a continuous surveillance among animals, or the sentinel areas where you find that these are hot areas from where there can be outbreak. And for this, as Joel said in the beginning, one health approach is necessary, where veterinarian, our clinicians, even basic scientists, ecologists like Umar sitting in audience, they should come and join hands in order to take up this task. Even I would say the universities which are situated in those local areas, even state universities, there are science department, they should also be approached to get involved in order to tackle this issue. So I would say effective surveillance is the only strategy.
With that, we come to a close. Let's thank Sunil for wonderful insights on